Okay, all right.
Hi, everybody.
Hello, welcome.
Thank you.
Ali is here.
Yeah.
Ali, I'm going to talk about you.
Hello, Javed.
Hello, welcome. Welcome to everybody.
Thank you.
Herkese merhaba. Bugün yeni bir webinarla karşınızdayız. Bugün sunum tam zamanlı araştırmacılarımızdan Doktor Javed Hüseyin Kolkar ile karşınızdayız. Bir taraftan tüm katılımcıların katılmasını bekliyorum hocam. Evet, şu anda bekleyen kalmadı. Güzel bir webinar olmasını diliyorum. Javed hocam, sözü size veriyorum.
Okay. Can I go?
Yes, please.
Okay, thank you very much. Thank you very much. Good morning to everybody. I hope you can hear me. My voice is a bit husky, so I have to push a lot of air with pressure to make it audible. I hope you can listen to me. If not, please give me a signal. I will try to mitigate my voice. I'm Javed. I did my PhD in India. For that matter, all education was completed in India. PhD in biochemistry, and then I flew to South Korea as a postdoctoral fellow on a national scholarship. I worked there for 5 years on biosensors and designing and developing biosensors, both genetically modified living cells as biosensors, as well as chemical sensors. Until 2009, in 2009, I came to Sabancı University to meet my future wife Anjum, who is also a colleague in this work, and initially joined Sabancı University as a visiting faculty in Faculty of Engineering and Natural Sciences from March 2009 until 2011. That's when the Nanotechnology Center was established. I got the chance to get into Sunum as a first researcher. At that time, there was nothing in this building, so we had to start from scratch. I'll tell you something about that one as I go on talking. From 2011 onwards until to date, I'm working as a researcher in Sabancı University Nanotechnology Research and Application Center. And a part of the research work that has been conducted is being presented in this talk. And the rest of the talk, I'll just give you a flavor of what all we did in the next slide. So to begin with, I like to show this slide. This sums up actually what we are doing at Sunum in terms of designing and developing biosensors. We had fantastic group of colleagues from electronics engineering over the years. We did a lot of research on developing an interfacing biological molecules with electronic transducers and went on to develop a handheld device. That's what you see at the left bottom corner here. That's the ready-to-use sensor, which is still under iteration, actually meant for detecting cancer and cardiovascular disease biomarkers. And before that, we had to establish a series of different designs, assays that go into developing this handheld device as a biosensor. As you can see, biosensor is a multidisciplinary area where you have to combine a lot of different disciplines to come up with a functional biosensor. So sensors that represent only electronic transducers, physical transducers, chemical transducers, and so on. But when you couple these sensors with the biological molecules, macromolecules, that's when we call them as biosensors that translate the biological signal into electronic signal that can be measured, or optical signal that can be viewed with our naked eyes. And now coming to nanosensor, this is what I'm going to talk about in this talk. We shrink down from macro to nano scale, essentially ruling out the role of these electronic and physical sensors that rely heavily on laboratory equipment. Essentially, you need a whole lab to conduct such kind of measurements. So I'm going to talk about that thing here. I'm not going in detail about this one, as you also will be seeing in my coming slides. This is the outline of the talk. I divided it into two parts. One is the general introduction of what these quantum dots are, and these their synthesis and their application. There are lots of applications. I'm going to highlight the specific application that we are interested in at Sunam, and how these quantum dots can be used in chemical and biological sensing. I will also highlight the major advancement that we made with respect to utilizing quantum dots in chemical and biological sensing, adding new functionality to these nano crystals. I would like to start with this slide. In a conventional biosensor, you got these components here. You got the electrical, optical, mechanical mode of transducers, where you utilize spectrophotometers, impedance analyzers, and measuring electromechanical features. All those require laboratory equipment that are based in the laboratory. You cannot take them out of the laboratory. And you, in addition to that, you need to have the signal processing system to amplify the signal in order to be visible or measurable signal. Because since we are dealing with the analytes in biological system, whatever the signal that is coming out is too small to be measured or too invisible to be seen. Therefore, we need to have this signal processing, signal amplification, and readout configuration to actually see the signal. What we wanted to do is that we wanted to get rid of these laboratory-based equipment. So in order to do that, we need to go down to the nanoscale. So what we do is that instead of having these receptors, maybe cells or nucleic acids or whole bacteria, yeast, or whatever it is, enzyme, receptors, hormones, antibodies, antigens, whatever you need it for sensing, you can have them coated onto small nanoscale crystal, nanocrystal. quantum dots are the better alternatives for going into the nano because of their fascinating properties, which I will be talking in the next slide. So as you have these quantum dots, they are not only optically active, now they are biologically active too. So you have two functionalities at a time. So you can probe the changes that occur at a nanoscale level. So once any target molecule like these ones, viruses, bacteria, or proteins, hormones, receptors, nucleic acids, RNA, DNA, and so on, any of those that can specifically bind here, and then shine the light on top of these nanocrystals, and the nanocrystals change its optical property. So they emit light of a radiation that can be seen actually with your naked eye. I'm going to show you what it means to be seen, these nanocrystals. I have that in my on my table here. I will show you in a few minutes. So what happens is that as soon as the target molecule binds to the receptor onto the surface, there is significant changes taking place with the emission profile of these nanocrystals. As you see, they are turned on before they are binding to their target molecules. When the target molecules bind specifically, the specificity comes from the embedded receptors on the nanocrystals. As they bind, if the molecule is sufficient enough to absorb this emitted light, you will see the light going off. That's when the signal goes off. So otherwise it is continuously on. This is always on type of sensor, nanosensor, that goes off only when its target molecule is captured. This is what we want. This is what we can, this is one way to eliminate the dependence on these laboratory equipment. So there are some molecules, for example, redox chemicals or redox drugs. There's redox enzymes that essentially interfere in this emission spectrum and influence shift in this emission peak. Such type of mechanism is often rare, but essentially they quench the emission of the light coming out of these nanocrystals. What are these quantum dots? Let me give you a brief fundamental background of these quantum dots. I hope most of you know already. These are semiconductors. They are semiconductors because of the band gap here. There is a band gap in all of the semiconductors. Band gap between the valence band and the conduction band. They exhibit quantum confinement. I will talk about this one in the next slide. They have energy band gap that determines required wavelength of radiation absorption and emission spectra. They absorb radiation from at one wavelength and emit light at a different wavelength. So this is all governed by the size of the nanocrystals. As you see in this one of the schematic illustration of a typical quantum dot nanocrystal, which is made up of core material coated with a thin layer of chalcogen materials that makes them more vibrant and brilliant color emitting nanocrystals. What is so special about quantum dots is the ability to have this property, quantum confinement. It is a spatial confinement of electron-hole pairs, often referred to as carriers, in one or more dimensions within a material. Take a look at this 3D structure here, 3D bulk material. In this material, all carriers act as free carriers, which means that they can move around in three-dimensional, different directions. While the planar materials of the same composition have carriers moving along the plane. So this is a typical property of semiconductors. So coming to the one-dimensional materials, often quantum wires, nanowires, where the electrons move into only one direction, down this direction. Quantum dots have 0 dimensions, and that's where the electrons and carriers are squeezed in a small confined place. That's why this system in which carriers are confined in all directions. That's what makes these nanocrystals so superior to these bulk materials. As you see, the bulk materials have a certain energy gap. When you shrink the size of these materials, same material, same composition, the band gap increases. That's what makes them highly unique nanomaterials. There are different types of quantum dots depending on the type of element that goes into. As you can see, this is the spectrum in which you can see different types of quantum dots having different optical properties. But we are interested, since we are dealing with biomolecules and trying to visibly see these quantum dots emitting light. So we have to deal with these cadmium selenide type of nanocrystals that emit light in visible range, that absorb light of somewhere in blue region or ultraviolet region. This is a schematic illustration of a core CDSC and coated with zinc sulfide, which is a popular combination. As you can see, the same composition of different size have different optical properties, which means that they have different absorption and emission characteristics. So as the size goes down, the band gap increases, while you have the constant source of absorption. So it absorbs light at this wavelength and emits light at different wavelengths, and that's why we see different colors. After having synthesizing these quantum dots, they need to be modified after post-synthesis modification. In post-synthesis modification, we can essentially cap these quantum dots with desired molecules. And there are a series of different chemicals that can be used to make these highly insoluble quantum dots into soluble quantum dots. That's the challenging task. Why it is challenging is that cadmium selenide is not soluble in water. There are other types of quantum dots called perovskites. They are made of halide, lead halide materials. They are highly water soluble and most popularly used nowadays for solar cells. Those have risks of getting into the food chain because of their high solubility. And CD is also a toxic material, but since this is not a water-soluble element, which is much safer than the lead halide perovskites. Quantum dots have been now used to harvest energy. They have been still in infancy stage. They are being used in photovoltaics. Bhavanti group at MIT is a very well-known group in this area. You already saw quantum dot TVs, quantum dots, TV display screens are made of quantum dots. I can see in the market nowadays, but their cost is relatively high compared to normal LEDs. That is because of their synthesis cost. Cost for synthesis of quantum dots is relatively high. And quantum dots have also been used in radiation converters, converting one color of radiation to another, which is a popular phenomenon. They have been used in this process. The quantum dots have also been used in various displays, fabrication of display materials using simple photolithography like the ones that is being carried out at Sunam normally with different materials, but that it is also possible to use quantum dots here. We discovered that two different types of quantum dots have been most popularly used in displays. One is green emitting quantum dots and red emitting quantum dots. These two types of quantum dots are often coupled with blue emitting LEDs that gives you red, green, blue, RGB. That these three colors can essentially be used in different proportions to generate 256 different types of colors. And if you use quantum dots, pure colors, vibrant colors can be generated. That's what quantum dots are famous for. How do they synthesize? How do we synthesize? Is this one. It's very simple. There are two methods to go for synthesis. One is organometallic type of synthesis, where you have to use organic medium in this one as the nanocrystals nucleate. These organic long chain hydrophobic molecules go on and cap on the surface of the quantum dots, making them more colloidal and dispersed. There are different metals that are being used. Cadmium in the range of visible. That's what I'm going to talk about today, with combination of zinc and chalcogens, sulfur and selenide. There are different types of metals that are used for different applications. As you go back and recall the spectrum, where you have different types of quantum dots used for different applications. I'm going to highlight only on cadmium-based quantum dots here. All that you need to maintain in the reaction is the flow of argon, because it is anoxic. You don't want oxygen to enter into it. If it enters, there will be no crystallization taking place. As soon as the precursor is injected into reaction, within 2 seconds, you start to get the crystals. We need 2-3 people to handle this reaction at a time, because if you delay, you will get a different size nanocrystal. This is one of the problems that we face while we were synthesizing in our lab. I'll talk about that in a minute. How to control the size is the main challenge. So there are two ways to controlling the size of these nanocrystals while synthesis. One is to take control of the precursors and timing of injecting the precursor into the medium is a critical task. Timing, temperature, and reaction conditions are the critical factors that govern the size of nanocrystals. In organometallic method of synthesis, essentially is a thermal decomposition reaction, decomposes nucleates, and then these long chain alkyl groups go on and cap the surface of nanocrystals. There's another method, aqueous, is more or less efficient method because the cuties that are produced in this reaction often have low photoluminescence and low quantum yield, which are not suitable for any application. So this is not very popular technique. There are different ways of synthesizing quantum dots like these, which is not a focus of present talk, so I'm not going to highlight on this. How do we use quantum dots as nano-switches? One of the best ways, the simplest way is shown in this slide. When you have these nanocrystals homogeneously suspended in a colloidal fashion, they collide, they don't settle down, they collide with each other and bump into the solution and remain colloidal all the way, no matter how long you keep them. But the problem is, if they are not surface functionalized, they tend to aggregate and settle down, and you can see with your naked eyes. This is one of the best examples where we can use nanocrystals. If you have a colloidal form of nanocrystal, this is one typical example that I found suitable to show you here. These are carbon dots, quantum dots made of carbon. These carbon dots, in presence of cerium-3, they aggregate and form a cluster that can be seen visibly. And the photoluminescence property changes. Photoluminescence property, emission property of these quantum dots changes as they form a complex like this, a coordination complex, which essentially quenches the fluorescent emission of the quantum dots, which we call the signal off. When in presence of pyrophosphate is a precursor for pesticides nowadays. That's why pyrophosphate properties is very well known in agriculture. When in presence of pyrophosphates have tendency to attract cerium ions and form complex, and the affinity of this attraction with cerium is more than the affinity of cerium to quantum dots, and that's when these quantum dots are set free into the solution and you have the signal back to normal. So this reaction can be used as a nano-switching reaction. So you can determine the extent of the analyte present here based on the amount of light emitted by the quantum dots, provided you control the concentration of the nanocrystals here. As you can see here, in normal conditions, quantum dots are clear. They start to emit light as you excite them. But when in presence of cerium, the signal is off. You essentially see no light coming out of the crystals. But when you put this pyrophosphate back into the solution, it comes back, it bounces back with emission of light. This phenomena was exploited in our study. This is a long time ago. Actually, the work actually started somewhere in 2013, and the work was published in 2015. What we did was we used yeast as a model. Yeast cell, the normal yeast that is used for fermentation, producing beer or something like beverages. So yeast is a good model in biology. Biologists know very well. Yeast has been used as a model for human cells to screen thousands of chemical compounds to rescue from disease in human cells. So it shares major metabolic pathways and homology with humans. That's why we need to study yeast to study human cells. What we did was we used yeast cells. We know how yeast cells divides and make babies, daughter cells. As you can see in this illustration here, we biologists know that yeast divides exactly in 120 minutes to give rise to 2 daughter cells. it's in biology, we call it as doubling time. So we know the doubling time of one yeast cell. It's very sensitive. If there is any external stimuli or stress that is coming in contact with the cells, this time changes, which means that some of the biochemical reactions that are taking place inside the cells are being slowed down or stopped. And that's what we can make out if the cell takes way too long time to divide. We use this phenomenon in here. One of the ways to study is to keep this cell alive, unaffected, and measure the signal. What generally, conventionally, people do is that they try to see the growth, time versus optical density, to see how many number of cells have grown in a defined period of time. But that is a very conventional method. You need a spectrophotometer to go and look at the signal. We wanted to get rid of that part. What we did was we had to go down and see the chemistry of the cell membrane, yeast cell membrane. And we discovered that this yeast cell wall is made of glucan and menon. And we targeted these disulfide bridges of glucan, which are vulnerable to reduction. And we also had the preliminary knowledge of the pesticides. The pesticides have this property to go and attack the living cells. How do? They do in various means. One of the ways is to reduce these bridges. Phosphate, organophosphate pesticides are very good in doing that kind of reaction. So we took one of the precursors of organophosphate, for example. This is TCEP. It's very popular. It's a highly strong reducing agent. It immediately reduces all the sulfhydryl groups, disulfide bridges like this one. So what we did was we briefly exposed these cells with TCEP, opening these disulfide bridges into two sulfhydryl groups, and that opened a channel for us to go and attack quantum dots, to attach quantum dots. So what we did was we took quantum dots with carboxy functional groups. Remember in the previous slide, we showed different ways of functionalizing the synthesized quantum dots. This is one of the examples out of that. We have a free carboxy group here, and we need a sulfhydryl group to go and attack these sulfhydryl groups generated as a after treatment with TCEP. These quantum dots then go on and form disulfide bridges here, and that's when they start to shine. We can control the concentration of quantum dots. We know the molecular landscape of this one, the area of the cell, and depending on that, we can control the number of quantum dots that can be decorated on the cell wall. What happens is that once we have these cells being decorated with quantum dots, now we know what happens. So we wanted to track what happens when the cell divides while these quantum dots are decorated on its surface. We know exactly that cell divides at 120, doubles at 120 minutes, and we wanted to trace the distribution of these quantum dots along the cell surface. This also happens on the human cell surfaces. What we discovered was that at 0 minute, as you can see here, this is the confocal images of the cells at different time intervals. As the cell go on to make daughter cells, this is the mother cell, it goes on making daughter cells at 230 minutes to 360 minutes, you have a complete division of the cell. So quantum dots can be traced to second generation. So quantum dots in first generation here, you still have same amount of quantum dots with some amount of being lost in the process of division here. And there are some traces of quantum dots in the mother cell, but not in the daughter cell. As soon as they become two daughter cells, you have all of the quantum dots disappeared, which means that there is a reorganization in the cell membrane taking place. And the debris from the mother cell goes on attached until the second generation. This is what we found out. Now, how can we use these as nano-switches? These are powerful tools to study the role of drugs, for example. There are so many drugs, anti-cancer drugs, malaria drugs, and so on. So we don't know what is going on. If you want to do testing, it's a time-consuming process. You need a lot of funds. You need a fully functional laboratory to do this kind of work. So if you use these kind of quantum dot decorated cells, yeast cells particularly, because it has a short division time, and compared to human cells, you can get the results very quickly and you can see it with your naked eyes. This is what we wanted to do. So we stopped there because we didn't have fun to carry on further. So we had to hold on to that part yet. But we established a tool that can be used for high throughput screening of drugs. If anybody is interested, then we can collaborate on that area. So we wanted to see what happens to the cell if the cell is being affected by the presence of quantum dots. We don't see any difference because the quantum dots are tethered or outside the cell. They remain outside the cell. They don't go inside the cell. That's one of the reasons why they are not toxic. People always apprehensive about the toxicity of cadmium in quantum dots, but we don't see that toxicity here. One of the reasons for that is because the thin layer coating of zinc sulfide on the surface of CDSC and the water insolubility of CDSC also contributes to the non-toxicity dimension. So we feel safe to be used in this one. One of the reasons also to not using in biology is because of the toxicity, that people talk too much about it. But there are ways to circumvent it. Pesticides are also toxic. They are being used to control, to increase the yield. They are being controlled. Quantum dots can also be controlled too. I will tell you a lot more about that part. These are images, confocal images. We wanted to try two different types of quantum dots to actually see if changing the size of the quantum dot does influence any property or viability of cells. We don't see any of that. That's a good sign. And then we went on to use it for testing toxicity. Why we entered this one? Because we got funding for this work. So we started to see the toxicity, nanotoxicity, using these nano-switches, living cell nano-switches. So we use different concentrations of these nano materials, double wall, single wall, double wall, multi-wall carbon nanotubes. There were, at that time, we had a lot of news around that time about the use and fascinating properties of quantum dots in electronics. We wanted to see what happens if we allow these carbon nanotubes to interact with living cells, which is not a good news though. As you see here, we discovered that single wall carbon nanotubes are more toxic because it collapses this cell structure. You will see in a minute in the next slide, followed by double wall carbon nanotubes. Multi-wall carbon nanotubes, although well, not so much toxic as single wall. This is another way of representation. You have different sizes, different types of carbon nanotubes here, increasing concentration from this end to this end. You will see the red ones are actually the ones that cause toxicity to living cells. This is actually the fluorescence coming out of the cell. If the fluorescence is normally coming out, then we'll have to assume that the cells are intact. Let me speed up because my colleague is warning about the time. This is how it looked like. This is single wall carbon nanotubes acting like nano syringes, essentially piercing through the cells, and all the cell debris is coming out as a result of cellular damage. And this is double wall carbon nanotubes, and this is multi-wall carbon nanotubes. They tend to accumulate at the junction of the mother and daughter cell. How do we see this one? We can actually see these reactions taking place here. As you see, under normal conditions, when there is no external stimuli, the cells start to emit light from their quantum dots on their surface. When the cells get damaged, there is no light because of the quenching taking place between the quantum dots on surface and the carbon nanotube, which is a well-known phenomenon. So double wall carbon nanotube, for some reason, did not cause toxicity as much as it did in single wall carbon nanotubes. This we can easily see it with our naked eyes. We also tried to do it with the bacterial cells. They are even faster growing cells. We can get it in maybe 5 to 10 minutes time. You can see these glass slide images, just like blood typing type of test. You can actually see it with your naked eyes. This is again the scanning electron microscope of quantum dot conjugated E. coli cells under normal conditions, and incubated with different types of carbon nanotubes here. And this is how we can see when we excite the quantum dots on the cell surfaces. And we went on to test with different chemicals. Paraquat is a highly redox cycling agent. In agriculture, they call it as pesticide. Hydrogen peroxide is a well-known redox agent. Triton X100 is a model for detergent. As you see, cells are not dying at low concentration of detergent, which means that it tells that we need to wash hands for a bit longer time to get rid of these bacteria. It can be seen here. So I will speed this process, coming to the part 2, which is to add additional functionality to the quantum dots. In addition to having this, making them biologically specific, optically active material, now we can add one more functionality, which is a magnetic property. So it gives unprecedented control over these materials. So magnetic quantum dot based immuno-optomagnetic point of care assay. This is what we developed in our lab, which is a patent application now pending. This is Ali. He's a very fantastic guy in synthesis of nanocrystals. You can ask lots of questions to him if you have any questions regarding synthesis. He's an expert nowadays now. This is me and Ali. Anjum is taking in the background because we need 3 people to handle this reaction. What we did was we wanted to control the synthesis of nanocrystals to atom precision, atomic precision. So we were successful in doing that. We used a lot of reported methods. None of them worked in our laboratory condition. Probably they worked in their own laboratories, but for us, only Bhavandi group work was very successful. So what we did was, in order to get uniform single color crystals, we had to have these quantum dots made of core, while the shells, thin layer of shells, should be in a gradient formation, layers formed in a gradient way. There is no clear-cut demarcation between cadmium selenide layer right on top of the core and the outermost layer made of zinc sulfide. So this was very interesting to us. We were successful in making. We can make in multicolors, but the demand came for only red and green. That's what people are interested in displays, while they use blue LEDs for emitting, and use blue LED to filter as well as emit red and green to generate RGB. So this is not the gradient one. You can have crystals. You can see there's even less than 30 seconds. We noticed that this comes around 5 seconds, even less than 5 seconds. We could get it at 30 seconds, which is already too late, which means that we need more people at the time of the reaction because it is not under our control. So this was the first attempt we got, and you can see the images here. These are in plastic tubes because we made them water soluble later on. Otherwise, this is not suitable to be in plastic tubes. Since they are water soluble, we had to use these fantastic tubes to handle micro volumes. After having this gradient synthesis, we could control the size and atoms in a nanocrystal. This is the single color for 4 nanometer diameter. You can see the size here, one of the sizes. And this is a little larger with 23 atoms, measuring about 7 nanometer in size. You have largest one. This is 30 nanometers in diameter, 30 atoms in diameter. Total size of 10 nanometer. That is one crystal. You notice these atoms here, they are same in size, but the number of atoms grow over time. As you see in the single color, this size essentially gave bright yellow. This is orange and red in color. This is all because of the band gap that we talked about in the beginning. That's another slide. So how did we made this water soluble is a very complicated story. To sum up this one, this is the tube showing nanocrystal under normal light. When you shine UV light, I have this one here. If you can see me. Can you see this one? Yeah. So that's the tube here in UV light. This is organic soluble. It remains in organic solvent, chloroform in this case. To make these quantum dots go into aqueous solution, we had to struggle really hard to use different linkers, chemicals, cross-linking agents, and so on to bring them to water. This is in water phase now. This is in Turkish because we use this for a report. And then we added another functionality to these quantum dots. We made them magnetic. As you can see in the top, you have videos coming next. I'll speed this one. 2 different types of quantum dots made magnetic. They can be separated easily. And then the reason why we added another magnetic functionality to this optical and biological functionality is because of their widespread application. This has not been explored yet, but you will see in coming years, you will have. Magnetic nanoparticles alone have been already in use for hyperthermia treatments, treating cancer patients, cancer tumors by using radiation. But if you have these quantum dots coupled with magnetic nanoparticles, you can actually see it once you excite it, as you can see in this case here. This is our area. I'm going to talk about this one now. I'll hold on to this one. These magnetic quantum dots can also be used as carriers of drugs, to carry drugs and precisely deliver at a specific location, both by using biological specificity and guided by the magnet. You can use magnetic field to guide these drugs to a specific location. You don't have to inject the whole drug into the body to spread. The whole applications can be seen in one of the recent articles that we published. Our vision was to use these magnetic quantum dots in detecting breast cancer. This is a patent application. It's recently submitted patent on this one. As you see in this video, one of the two quantum dots are made magnetic while the other one is non-magnetic. So you can essentially determine the presence and the quantity of a biomarker from a serum sample. So I'll skip that part. Coming back to our invention, what we did was, we as I said, that we didn't want to have these laboratory equipment to measure the signal. Then how can we measure other than by seeing qualitatively? Seeing with our naked eyes is only a qualitative method, but if we can measure, it's more quantitative. So how can we do that? Everybody has cell phone in their pockets now. If you download an app and just point it to the device and take a picture and subject it to processing using specific algorithm. In this case, we utilize RGB profiling algorithm, red, green, blue, which will display the ratios of red, green, blue in a given image, as you can see. This is a real-time video taken at the time of the measurement. You can see the changes in the values that can be correlated to the concentration of biomarker present in a solution. And this process is a very complicated process. You can get a sense of what it means like to develop a sensor and then measure. So skip this one. How did we make this one? Is first we had to synthesize a very precise size controlled magnetic nanoparticles. We employed density gradient centrifugation as we commonly use in biology. Sucrose gradient, cesium chloride gradient centrifugation. You build up the gradient of concentration of these sucrose or something like that, and then builds up the concentration. And then when you pull down using a centrifugal force, relative centrifugal force, with the different gradients of concentration of these sucrose or cesium chloride, you can essentially control or separate or isolate specific sizes. We were interested in precisely controlling 10 nanometer size. As you see in the SEM image, TM image top here, it's precisely, roughly, not precisely, close to 10 nanometer, which is what we wanted, because we wanted to keep the dimensions of our nanocrystals, quantum nanocrystals, and the iron oxide nanoparticles identical in their size, so as to make them fluorescent, as good as quantum dots do without having these magnetic nanoparticles. This video shows the difference before and after coupling quantum dots with magnetic nanoparticles. The right hand side is the same tube as you see here. This is magnetic nanoparticles. The left hand side is the one magnetic quantum dots actually. This is another video that shows we had to control the orientation of the quantum dots and the iron oxide nanoparticles in a way that quantum dots should be buried inside while the magnetic nanoparticles should be surrounded on the peripheral part of the quantum dots, making these quantum dots essentially hidden inside a complex architecture. As you see here in EDX map, the quantum dots are located at right at the middle of the nanocrystal, while iron oxide nanoparticles are being protected along the surface. Okay, let me go back. Yeah, so the whole process this was shown previously. Just to remind you, you have a drop of blood. All that you need is a drop of blood to detect the biomarker. In this case, cancer biomarker, especially breast cancer. You have magnetic quantum dots here, conjugated with specific receptors. Here, in this case, anti-ERBB2, anti-HER2 antibodies, specifically bind to HER2 protein, which is a biomarker overexpressed in breast cancer cases. So the having magnetic property of these magnetic quantum dots allows us to pull them down from a dilute solution, concentrate it, and then apply it onto the surface, which has another type of antibody, but binds to a different epitope of the same antigen carried by these magnetic quantum dots and analyte complex. And that's when we get the signal. They strongly stick onto the surface of the chip, and washing this one will leave you behind the complex, and we can calculate the concentration. I have this chip still with me here. This is how we did. So this is the image showing, and all of these components go into an assay kit, and this assay kit is essentially used, can be used, potentially be used for detecting breast cancer. Otherwise, you have to go through all of those processes that I mentioned in one of the slides. So look at the magnitude of complications that one has to go through to detect these things. By the time it is already too late, what if we have a technology, an assay method that allows a woman to readily detect this even before the signs emerge? This is the whole purpose of our work. Let me speed this up. This is another image. There is another image here, video here. This is the concentration dependent responses. As you see this heat map here, 3-dimensional heat map that essentially shows the increasing concentration of the cancer biomarker in the sample. By the way, these are the authentic samples, model samples that we use, spiked into the serum and then measured the concentration. I'll skip this part. There is huge market demand for such technology. And where else we can use these quantum dots? In clinical diagnosis, in R&D laboratories, detection of viruses, bacteria. We applied for funding for virus detection, and hopefully we should receive it, probably. And other potential areas could be pharmaceutical industries, environmental agents, defense agents, agriculture. I didn't mention here. One of the future aims is to use this in agriculture to remotely sense and monitor agricultural crops and fruits. I think I should stop now. The materials that we used here in our study are now made available in our e-store. If anybody is interested, we can go and look at it if they can find that material from website. We are a very small team, very dedicated team. I and Anjun, we have 24/7 discussion time, unprecedented, which is hard to find such things. That's one of the reasons why we couple all the time. You'll see some of them are annoyed, but it's working for us. Ali is a fantastic person that came into our group a year ago. And now he already is expert in this field. Former members are Raguraj Chauhan, who was here for 3 years, Ashish for 4 years. They did a wonderful job. Thank you very much for listening.
Javet
Are there
Hocam,
any questions?
Javet Hocam, thank you very much too. Katılımcılarımızın sorusu var mı acaba? Do you have any questions? In chat box, somebody's thanks to you, Javet Hocam.
Thank you. Thank you for listening.
Peki tüm katılımcılara çok teşekkür ediyoruz. Haftaya bir başka Nano Open'da görüşmek üzere. Javet Hocam'a da tekrar ayrıca çok teşekkür ediyorum. Değerli sunumu için çok kıymetli bilgileri bizimle paylaştı. Katılımcılarımıza da çok teşekkür ediyorum. Haftaya görüşmek üzere. Hoşça kalın.
Thank you very much.